because this question will going to define the phenotype of a cell, the biology of the cell. This will going to define the biology of the cell, that whether a cell will going to live a life like a normal cell, or it will going to live like abnormal cell, or a cell with a disease. But let's be more positive. The outcome of this regulation will define that whether this cell will going to live like a kidney cell, or it will going to live like an eye cell. So the difference between different cell types is embedded in the ability of the cells to regulate its different gene, is the expression of different genes. And the first and the foremost way of controlling the expression of the gene is actually hidden in the cis-acting DNA sequences. Let me be a little bit more traditional to answer you about this thing, the cis-acting elements. Okay, we are all set for that thing. I think हम सब लोग पूरे हो चुके हैं. We can stop. We can start without much problem. Okay, all set. The first thing you should know is, can you see the my notepad?
Yes, sir.
Thank you very much. Okay, the first thing you should know or recall once again is about the general structure of the gene. Every gene is supposed to start from a place which is called as TSS. It is called transcription start site. Whatever comes after that is part of the gene. And you all know that the RNA pole 2 binds here along with the transcription factors, and then it produces what we call as messenger RNA. That messenger RNA will eventually ended up into the protein. That's the old story. I am not going to spend time on that thing. Whatever the length of the DNA present above the transcription start site, all that portion is critical, is very important to plan or to make a decision whether this gene will be expressed or not. Another important thing you should know, that the RNA pole 2 is a big molecule, is a big protein, I must say. The RNA pole 2, they don't have eyes. They don't know where to start. They don't know whether to express a particular gene or not, whether to start the process of transcription or not. RNA pole 2 have, like, they don't have any things like eyes. They cannot make a decision. If the RNA pole 2 is present in a perfect working condition, and I can tell you that one of the perfect working conditions is like somebody puts a fire on the RNA pole 2. And that fire is, I must say, it is phosphorylation. Is phosphorylation. So once the RNA pole 2 is stick with a DNA strand and it gets phosphorylated, it like boom, it can start running from here to there, and then it start producing messenger RNA. So after that, there is no way you can stop the RNA pole 2 for the execution of its function. That means for the production of messenger RNA. The gene will be expressed after that. And what is the active functional conformation of RNA pol 2, which you can see that it stick with the place just above the gene and it gets phosphorylated. The portion where the RNA pol 2 will start its assembly procedure, that portion is called as promoter. This is called as the promoter region. A promoter is further more divided into different subfragments. But first thing you need to remember, this is the place where the expression of the decision about the expression of the gene is made. Certain proteins came and bind themselves to that place. Certain proteins came and bind themselves to this place. All these proteins are famously called as transcription factors. Please remember all those things. They're very important. They're called as transcription factors. They are proteins. You might recall that those are the proteins which help in the expression of the genes. They help in the expression of the genes in different ways. One of the ways that they detect certain sequence of DNA, which is called core promoter, which is called core promoter region, and those proteins bind themselves on that region. So the proteins have the potential, has the ability to bind themselves on the core promoter regions. And you can guess the core promoter regions are present just above the gene, somewhere here. They created the platform which allows the recruitment of other proteins. And that combination will allow the recruitment of the final player, which we call as RNA pol 2. And those proteins not only help in the recruitment of RNA pol 2, but also phosphorylate this RNA pol 2 so that it can proceed with the synthesis of messenger RNA. This is one of the ways that transcription factors help in the gene expression. Those transcription factors are also universal. They are present in every other cell. There are certain other transcription factors which helps in the recruitment of those proteins here, which indirectly help the expression of the genes. There are certain transcription factors which are directly helping the RNA pol 2, and there are certain other transcription factors which are indirectly supporting those transcription factors so that it can proceed with the expression, it can proceed with the transcription. So as a molecular biologist, you should be very much clear, very much clear about the different fragments present in the promoter region. It's a key concept. Everybody will expect from you that you have clear understanding about all those different regions present in the promoter. Okay, there is a message. Okay, I have a message that video बार-बार disconnect हो रही है. बाकी लोगों के लिए भी हो रही है. It's just for one person. The other students are getting me comfortably. Please answer.
Yes, sir.
Okay.
Yes, sir.
Okay, thank you very much. Then it's fine. वो जिस स्टूडेंट का मिस हो रहा है उसको वीडियो मिल जाएगी बाद में. I mean वो इंटरनेट कनेक्शन की वजह से हो रही है. Okay, let me take you to the first type. Okay, first thing, the first portion on the promoter. Now you can have an idea where the promoter region exists just upstream. In molecular biology, it is called as upstream region, just above in the upstream region. Can you see, for example, if this is the gene here, Let me help you by highlighting those things. So if this is the gene as I have shown you, this is the gene, इन्होंने अच्छी पिक्चर बनाई है. This stuff, ये जो आयरन की तरह का ना, इस तरह की तरह का stuff, this is the RNA pole 2. Okay, this one is RNA pole 2, and this will supposed to produce the messenger RNA. Okay, so this is the RNA pole 2, and you can see the other things, the other proteins. These proteins are critical. They are the transcription factors, TF2s, they're called as TF2s, and there are others. They created an environment, they bind with the core promoter region, and they allow the RNA pole 2 to be recruited at that place. Imagine RNA pole 2 is all over there within the nucleus. But RNA pole 2 is not expressing, is not transcribing every gene. RNA pole 2 is not able to bind with the DNA until unless this setting, this type of configuration was not set. The other proteins create like, they create like a structure which can hold the RNA pole 2 here. And after that, you will see the tail of the RNA pole 2 is get phosphorylated and it start proceeding with the process of transcription. It's almost a similar type of things I have shown. The first thing, the first part of the promoter region is called as the core promoter region. It's very critical, very important. The core promoter sequence contains the Tata box as well. If you can go back to the paper which I shared, my paper, if I claim that a certain portion is a core promoter sequence, I should also require to show the Tata box over there. The Tata box is a TATA sequence. This sequence is always present within the core promoter region. It's typically located near the gene. Upstream means upside of the gene. Downstream means just beneath the gene. And you need to remember the direction of transcription, okay? Upstream means upside of the direction of transcription. Downstream means after or towards the other side of the direction of transcription. This is the place where TF2s means transcription factors and RNA polymerase actually bind and connect with the DNA. And whatever the structure they create, that structure is called as TI complex or transcription initiation complex. The TI complex, if created, it can proceed with the transcription and it will give you a minimum level of expression of that gene. It's called basal level of transcription or constitutive expression of the gene. So you will be able to get constitutive expression of gene if this setting is established. That setting means the TF2s are here and RNA pole 2 is here. So what that means? Now that gene will be expressed like at a minimum level or at a basal level. The next portion is called proximal promoter region, or the indication of that region is with the presence of GC box. It's located immediately upstream of the core promoter sequence. So if this is the core promoter sequence which recruits the TF2s and RNA pole 2, the proximal promoter region is present upstream of that thing. I wish that you should have enough thirst in your head, not thirst in the stomach. If you have enough thirst in your head, you should go back to the sequences of the genes. You should mine the sequences of the gene and try to find out that there is the transcription start site. This is the gene. This is the core promoter region. This is the Tata box. This is the proximal promoter region. This is the GC box. You should try to see all those things with your very own eyes. Okay, so everything upstream above the gene is called as the upstream sequence. Okay, this is the upstream, and from here the downstream. Here the transcription starts. So this is the core promoter, and this is the proximal promoter region. Now the third one is called as the enhancer sequences. The enhancer sequences can be present. It's usually present far away from the gene. It can be present upstream of that gene. It can be present downstream of that gene. But the enhancer sequence increases the expression of that gene. Earlier, it was like a constitutive expression, basal level. If the TI complex is there, transcription initiation complex can produce, the constitutive expression. But with the presence of an enhancer sequences, the expression is increased a lot. I will show you how a sequence present far away is able to increase the expression of a gene. So there are different elements present on the same stretch of the DNA. All those things collectively are called as cis-acting DNA sequences. Anything which can influence the expression of the gene present on the same stretch of DNA is called as cis-regulatory element or cis-acting element, sometimes cis-acting DNA sequences. Somebody asks you a core promoter region, this is a cis-acting DNA sequence because it is present upstream and it can influence the expression of the gene. A proximal promoter region is a cis-regulatory element because it is present upstream on the same stretch of DNA. And then all of those things are also cis-regulatory elements. Let me explain these small things. The first, enhancer sequence. Remember, enhancer sequence is the portion of the DNA. It's a very small portion of the DNA. But luckily, that portion can bind a certain protein. We don't know XYZ protein. Here, the GRP is written just for an example, but it may be any other protein. So enhancer sequence is able to bind itself with a certain protein. Due to the configuration of DNA, although this portion is far away from DNA, am I right? This portion is far away from the gene. यहाँ से gene शुरू हो रहा है. The gene starts from here. And then there is a core promoter region, then there is a proximal promoter region, then blah, 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 blah stuff. And then after long time, far away, there is a sequence called enhancer. It's supposed to be very far, but because of the three-dimensional architecture of the DNA, this is actually present in a very close vicinity of the core promoter region. And also it can bind with another protein, and that protein can trigger, can kick, can push the transcription initiation complex to proceed at a faster rate. And that is why enhancers eventually able to push the expression of a certain gene. This is important. Let me explain this thing in a little different way using that. So for example, here is a thread of the DNA. I am just making a break here in between. So that means there's a large distance between these two. Here is the gene. So whatever the region present before the gene is called as promoter. You can guess core promoter should be here. You can guess proximal promoter should be here. And then there is this sequence here. This sequence is enhancer sequence. Now you might wonder that this is present at a very far away. How this sequence, this enhancer sequence is able to influence the expression on that gene. Okay, let me draw, allow me to use the my notepad. Okay, sorry, you need to switch between these two things. ओके अगर आप मेरे साथ हैं तो थैंक यू वेरी मच. अगर आप कम है साथ तो फिर भी ठीक है, कोई बात नहीं. मैं नहीं तो अकेला लगा होता है. आपकी मेहरबानी, आपकी वजह से. Okay, let me show you that how a distance present far away is still able to control the expression of the gene. Let me redraw the things. This is the gene, this is the promoter region, and then far away, this is the sequence. This is an enhancer sequence. It is an enhancer sequence. हमें कैसे पता लगा? This enhancer sequence contains a sequence of DNA which can bind a certain protein. For example, this is the protein X. I don't know why I put always X. X-Men अच्छे लगते हैं मुझे. So this is an enhancer sequence, and this sequence is supposed to affect the expression of this gene. If the TF2s are here, if the RNA pol 2 is here, so this thing is called as TF2s and RNA pol 2 is here. If RNA pol 2 is here and TF2s are here, what that mean? This will going to produce a basal level of expression, and this is called TI complex, transcription initiation complex. Now this will going to produce a basal level of expression. How it is possible that a DNA sequence present far away, kilobase pair away, and still able to influence it? We cannot understand this thing until unless we know the three-dimensional architecture of the same gene. When scientists able to understand or resolve the three-dimensional architecture, they came to know that actually the stuff which is supposed to be far away is actually present very near to the core promoter region of that gene. RNA pol 2 is here, TF2s are here, everything is here. So the three-dimensional topology, the three-dimensional architecture of the sequence of the DNA brings this portion very close to the TI complex, and that also brings that protein near to the promoter region, and that protein can kicks the TI complex to start producing a large number of messenger RNA. Now guess, it's a miracle. There is something, a sequence of DNA here present on the cis side of the DNA. If that sequence is here, it will recruit this protein. Because of the three-dimensional topology of the DNA, that portion comes very close to the core promoter region, and it will bring that protein very close to that core promoter region, and it kicks and produce large number of messenger RNA. Further, let me complicate the things for you. If in one type of cell, if in a given type of cell, if the cell does not contain protein X, if that cell does not produce protein X, what will happen? If I ask you whether the enhancer sequence is there, the answer is yes, of course the enhancer sequence is there, but the cell is not producing protein X. That is why the cell will not be able to kick the transcription initiation complex very well, and they will not going to produce that much messenger RNA of this gene. What if that enhancer sequence got mutated? What if the enhancer sequence got mutated? It will no longer able to bind with this protein X. and that is why it will not going to not going to push the expression of that gene as well. There are lots of possibilities are out there. Only this functional conformation results in the expression of this gene in that much quantity. In every other state, that gene will be expressed in a basal level. And this cell will going to produce a cell type A, and this cell will going to produce another type of cell called the B. And this is a really special and wonderful understanding we have developed over the period of time, that how a cis regulatory element can control the expression of the gene. Now just add more complexity, but with the same concept we have just generated. There are another type of sequences, cis regulatory sequences. One of them is called as silencer. ये आपने सुना होगा वाले लफ्ज किसी ने?
Yes, sir.
कहाँ? चिल्लर पार्टी में सुना है ना?
Yes, sir. वो ज्यादा
चिल्लर पार्टी,
बाइक्स का silencer.
बाइक का सलेंसर भी होता है। हाँ, चिल्लर पार्टी ने देखी आपने, एक इंडिया की बच्चों वाली मूवी है, उसमें ना एक लड़का है, वो जब भी बोलता है ना वो पनौती उल्टा ही बोलता है, तो फिर उसको ना उसको नाम देकर चुप करा कर दो, साइलेंसर. So silencer sequence is just any DNA sequence. It's just one type of DNA sequence. Remember, we can refer a DNA sequence as enhancer if it is pushing more and more transcription, if it is supporting a gene for the expression. We can consider a sequence of DNA as a silencer sequence if it slows down the expression of a certain gene. So let's suppose there's a sequence of gene called silencer. It can recruit another protein called GRP. You can refer any XYZ protein here, okay? It's a regulatory protein. It binds another type of regulatory protein, but this regulatory protein can actually block the function of RNA pol 2. It can block the function of RNA pol 2. Now guess what will happen this time? If the silencer sequence is there, it can recruit a protein, and because of the three-dimensional structure of DNA, it will bring that protein near to the core promoter region, and it will block the function of the transcription, slows down the function of transcription. A similar sort of work, a similar sort of blockage is offered by insulator. लेकिन ये जरा बड़ी बात है, इसको फिर कभी देखेंगे, insulators. And then there are certain other elements called response elements, and response elements are those elements which can act, which can affect in the presence of a certain chemical, in the presence of a certain chemical. हम वो पढ़ेंगे वो chemical कौन से, उनको बोलते हैं response elements. So all those things are called, in totality they are called as cis regulatory elements. I hope you understand these things. They're called cis regulatory elements. Now imagine when scientists are working on these things, they need to prove that this protein binds here, and after that it is interacting with that protein which actually connects with an enhancer sequence, and that is why it is an enhancer sequence. This is a silencer sequence because it binds with another protein, and because of the three-dimensional structure, it brings that protein near to the co-promoter, which blocks the function of the RNA pol 2, and it decreases the expression. We need to prove all these things scientifically. Okay, and I have proved a certain or a similar sort of stuff, which I will explain later, maybe. And then the silencer, then the insulator, and then the response element. The response element sequences are important because there are many response element sequences out there. They are short sequence of DNA, usually present upstream of the core promoter. Pakki baat hai, they should be present upstream of the core promoter. They can change, modulate, or regulate the level of the expression of a certain gene. One thing you should never forget, if the transcription initiation complex is here, this is, if the transcription initiation complex is here, if this stuff is created, a basal level of expression will be there. If this thing is supported by an enhancer sequence, the expression will be increased. If that stuff is stopped by a silencer, the expression will be decreased. So the expression, the basal level or constitutive expression will be enhanced or will be decreased depending on the nature of the DNA element interacting with the core promoter. Core promoter ke paas kaun aa raha hai, okay? And one of the DNA element which can regulate the expression is called as response element. The response element are short distance upstream of the core promoter. They present upstream of the core promoter. They are short sequences of DNA which can get activated in the presence of certain stimuli, in the presence of certain chemicals, in the presence of certain growth factors, interferons, heavy metals, CAMP, cyclic AMP, heat shock, steroid hormones. All these factors can affect those response elements. Since there are many factors, so there are many types of response elements as well. Please be very clear on this understanding. Let's suppose the first one called CRE. So if there is a CRE here, CRE is called as cyclic AMP response element. So RE will always be response element. The first is the name. So CRE, cyclic AMP response element. Remember, actually it's a sequence of DNA. It's just a sequence of DNA, but that sequence of DNA can activate when a cyclic AMP is there. Okay, so there is a gene. If that gene is associated with a CRE element, in the presence of cyclic AMP, in the presence of cyclic AMP, that CRE region will be activated and it will push the expression of that gene. How? Just see here. So this is a response element. If cyclic AMP is present, it can bind itself here, it can push the expression of that gene. But that gene will only express once the cyclic AMP is there. If the cyclic AMP is not there, the CRE will not be activated and the gene will be expressed in a constitutive way, in a basal level expression is there. But in the presence of CAMP, it will express more than the basal level. Another factor, serum growth factor response element, important one. But that will got activated in the presence of serum growth factor in the same way. Interferon gamma response factor or IRE. If a gene is supported by IRE, means interferon gamma response element, that gene will got activated if the system contains interferon gamma response element. A very interesting stuff is HSRE, the heat shock response element. Let's suppose there is a gene in the same way, and here is a DNA element which is referred as HSRE. If this is a heat shock response element present in a gene, upstream of the gene, what that mean in a simpler words? That means if there is a heat in the system, if there is a heat in the system, if a cell of a structure absorb a lot of heat, that stuff will activate it. And activation of heat shock response element will trigger the expression of that gene. Otherwise, that gene will be expressed in a basal level. But at higher temperature, the heat shock response element will push and trigger the expression of that gene to a large extent. So there are certain genes which are expressed when the cells experience a sudden heat shock. And that is only possible because of this. So all these response elements are very critical and important. Okay, so this is your task you need to do. There is a gene called PAX3, a very famous gene, and you need to explain to me that how the PAX3 is regulated. One way or another, whatever I have taught you, all these things will be contributing in the expression of the PAX3. So it's a real example out there. It's a very famous gene. You will see there are a core promoter region out there, a proximal promoter out there. You will see, can you guess this arrow? This is the transcription start site. Okay. So whatever present above here will be, there will be a core promoter region, then there will be proximal promoter region, and then you can see the distant elements, maybe enhancers, maybe silencer or blah blah things. There will be transcription factors. When you Google them, Google their names, you will come to know. Okay, if there is an arrow, I don't know why I'm telling you, this is your task, but anyways, if there is an arrow here, that means activation. So this thing is supporting or activating. If it ends like this, that means stopping or blocking. So SHH, sonic hedgehog, is name of the protein. It blocks NCE2. Okay, I am not supposed to tell you. Okay, one more thing you should know, that the core promoter region or the promoter region is not always supposed to be present in the upstream of the transcription start site. Sometimes it is present within the gene. So regulatory elements can be present within the gene like this. It's a very interesting point. You should remember that thing as well. Okay.